My question is for ViaCyte. I've heard conflicting viewpoints on whether or not a preliminary interaction with the FDA is a good thing. Could you just elaborate on your experience with the FDA while you were planning your preclinical studies?
Sure. So it's been a long interaction. And I've joined this company only in the last 4 months, so I can't tell you exactly when they did their pre-IND. But I can tell you this. It's been a very interactive relationship. There's been multiple meetings, continues to be multiple meetings. They've been, I think, CBER, the point group in this is CBER. We're developing a combination product. So CBER is the primary group because you have a biologic, but then the device group comes in an advisory role. And I think you could ask anybody in a company, I think you'd get a consistent answer that it's been a very collegial relationship. It's a very early space. I think CBER recognizes that there's many issues and challenges. It's a very difficult product to develop. I mean, these are very, complex products. Just the cell therapy product alone is complex, and you layer in a device, it's even more complex. So I think it's been a very positive relationship, and they've been very engaging with respect to advice. I can tell you just recently with that discussion with the FDA in February, the team went in, discussed all the data that we had to date, put a plan on the table, and there was very quick alignment with respect to a preclinical plan for this next product. So I think it's been very collegial and positive.
Your question over here, Barry.
You just keep jogging back and forth. I need my exercise. We're right in front of the cameras.
Oh, sorry.
Yeah, please.
So my question is actually also for Dr. Zimmerman. I didn't quite grasp, you were saying one of the things you did to improve your latest product was to increase the porosity of the encapsulations so that you could improve vascularization. I would have thought that once you have vascularization of the encapsulated product itself, you'd immediately get the autoimmune attack that is the main reason for doing the encapsulation. So what's the story there?
So with the porosity, when we add the pores, you do have to use immune suppression drugs. Okay, so that is essentially, the PEC Direct really solves half of the challenge, right? There's two challenges. One is the cell source, and the second is the immunological challenge. So the PEC and CAP, the intent there is to solve both of them, okay, and for a very large patient population. And the PEC Direct solves essentially the cell source issue, which it provides a scalable cell source. It provides a QC'd cell source that's consistently made, and under GMP conditions. So, but you still have to use immunosuppression. So again, it's not going to be for every diabetic, but it's going to be for the diabetics with the greatest unmet need.
Are there any other questions? Oh,
There's one here,
front.
but meanwhile, let me ask a question. Bernard raised the point of, so with the increasingly instrumented patient, if you will, we learn more and more about people as individuals as opposed to groups of people. And so you can sort of start to imagine, ends of one when we're thinking about how do we treat diseases. And Khalid, you were starting to go edge down that path when you talk about, well, those we operate on, some aren't going to be responsive to trail, blah, blah, and you looked at people, different tumors responsive to different treatments. And Mark, I know in the diabetes side, not all diabetics are the same. And they're not only the other conditions they have, but for example, there are cohorts of people with diabetes who've lived successfully very long, lifespans, right? Mm-hmm. And are sort of less impacted by the disease than others are. So I guess, collectively, how do you think we're going as we look at disease, whether it's sort of, brain cancer, diabetes, whatever, as we go down to sort of increasingly narrow segments of patients? And in fact, are some of the solutions you're talking about today, going to be relevant, say, 5 years from now, as we know more and more about increasingly narrower segments? And then, so I'll ask you guys to comment, and then Bernard, say whether you think they're right or not.
This is actually a fascinating question. I'm not sure we have the answers. I think what we're trying to do ultimately is reconcile precision medicine with fuzzy biology, which, as hard as we push, I think ultimately will keep a certain degree of fuzziness. I mean, you know, the concept of my genome is obsolete. I mean, we know that we're a mosaic of various genomes. If you sequence various cells, not even if you're not cancerous, if you sequence various cells, you'll find, you know, different genomes that vary to a certain extent. And what does that do to you know, our biology is unknown, and I'm not sure it's knowable. So there is, you know, the blueprint is fuzzy and will remain fuzzy, and our extent to intervene precisely in that very complex organism, I think, will have some limitation. And we don't know yet what it is. I think we've got to explore. But that's my concern. I mean, precision medicine is a great idea, but I think it will have some limitations.
Yeah, I think personally, so I don't think you have a one sort of strategy for all diseases. For example, the tumors in the brain, I don't think anybody can predict somebody would get a tumor in the brain. So you can't put a device on a healthy individual and say, I'm going to monitor that, you know, you will have a proliferative cell in the brain. You can do that on a patient that has had a breast cancer previously and has a potential to develop a metastatic tumor later on, that you might want to put a device, look at the circulating tumor cells, or monitor circulating tumor cells every month to see how many there are. So I think it will vary from disease to disease. I don't think there is one big strategy for all. I think that's my perspective from a more of a cancer perspective. But I think, you know, Mark can add to it from
I do think, though, to your point, analytics and biomarkers and diagnostics are going to play a very key role in future medicine. You know, so the ability to predict who's going to respond, who's not going to respond. And you hear that already. You know, payers don't want to pay for something if, you know, only 40% respond, right? So I think the idea, I think that's a very robust area, and I think it's going to be, and reimbursement's going to be a big part of that also.
Let's come back to the reimbursement question, but I know I interrupted a question over here somewhere. Yeah.
Yes.
Well, I interrupted Jerry walking to you, sorry.
Stopped me. Hi, my question is for Dr. Shah. I found it very interesting when you were talking about taking your therapy out of the lab and potentially into clinical trials. You said that you had to turn it into a company. And additionally, you expressed a bit of discomfort about balancing the academic side of what you do and running your company. And then when you were talking about taking R&D to clinical trials, you said that there's a lot of issue in the interface in between turning something from R&D to clinical trials, and that academia doesn't have the skill set to do it. Do you think it is possible that it was a correct, I guess, modification of education of academics, that it is possible for academia to push it all the way through to clinical trials? Or do you think it's necessary that it has to be industry collaborations to bring it all the way through?
That's an excellent set of questions. I think it sort of sums up. So I think what will happen and what has to happen in future, that of course you will have to have people with certain expertise. You can't be a sort of jack of all trades and try to take it, say, okay, I innovate and I will go to the clinics and treat a patient. It's possible, but I think, you know, what I meant is that you got to find people who are going to unite the people who innovate, the people who you know, actually work in the clinics, and the regulatory affairs people who will basically sort of approve your therapies. I was not uncomfortable with it. I said, but it's as a researcher, you basically, your life revolves around, and I think many can vouch for that, that your life revolves around getting grants, publishing papers, and hiring people. This is the cycle of a researcher. And you have so much stress in this whole circle that if you miss any of this, your wheels are going to be off. And I think we know in the current funding situation, that's a real fact, that if you deviate from this, you're going to lose the funding and your research will fall off. Now, the question is, what effort does it take? And then you have to raise money. And the point you made is that, is there a comfort zone? Because you will have to raise the money in the academics to take it into clinical trials. And nobody is going to give you $20 million in the academics unless you patent your technology, give it to the licensing authority in your university, and they're going to bring in the money. So that's what we did. We basically had the patents, licensed them to the company that we founded, and that's when the angel investors came. They were not going to give the money to my institution because the institution was going to take 74% of their overheads, which is enormous amount. Why would they give 74% to the institution and not make the company to move further into the clinical trial? So there are some, pluses and minuses, but what I meant was that you need personnel who will basically connect all the three and make the triangle what I just showed. And I think educating people in academics how to create companies and then to move them into the next level is the key. And I think there are people there, I know a few individuals. One of them is Glenn Prestwich, who has done this at the University of Utah. He's created 60 companies, and he's sort of educating people how to do this regularly. And I think what you have to have on board is the people in the licensing department of your institution who are going to work with you. Because before we made the company, they were not going to patent everything. Now they want to patent even a drop of water from my lab. So no, this is the fact. I mean, so they see the potential that is going somewhere meaningful.
Really good water.
Yeah, good water. Yeah. It's Poland Springs.
We have a drought in the Northeast right now, and we have a question over here.
Thank you. That's really awesome. My question is, I know more and more we hear a lot of China doing incredible stuff when it comes to genomics, synthetic biology, how they're going to actually, as a country, advance some of the developments that we're right now speaking about. From your vantage point, in each of these disciplines, either targeting a disease or just overall looking at bio as a mechanism to somehow manage your current physical body and maintain it in better health, attack the disease, also increase longevity. How do you see the debate around designing humans in a synthetic lab environment? And is there something would you be promoting, both from an ethics standpoint as well as from a medical standpoint, try to advance for the sake of science, regardless of national policies, everything else? So how do you personally see yourselves playing a role in continuing to focus more on not just reading the genomes, but actually writing the genomes and designing the genomes for better human capacity in the future?
I don't know if I have an answer to that one.
Brock might be in a better position to answer this.
Yeah, I really don't understand fundamentally your question. So can you?
The question really is, if you leave the ethics aside, we are now developing, biological tools that can better edit, monitor, design at the genomic level. And
sure.
So if you have to purely go into the design phase of it, not just edit something out, but designing from scratch, how would you look at that? If you have to design a human body in the future, how would you look at that in a lab environment?
Okay.
Or from a research environment?
Now I can take a shot at it. Well, I think it's an extremely exciting time. I mean, if you look at the tools that have been developed just in the last several years, they didn't even exist before. You know, it's just incredible, the molecular biology. It's almost exponential as far as, and if you look at it over a decade or so, or it's even more boggling. So, you know, I think at the end of the day, there's opportunities to just develop just really interesting and novel products. So for example, especially in the regenerative medicine space, we've talked today about, you know, the issues of autologous therapies. Certainly from a business model perspective, allogeneic strategies are something that I would say companies can get their arms around a little easier, and there's a business model for that. And with the technologies today, you could potentially edit these cells from an immune perspective, whether it's a cardiac cell or a diabetic cell or a Parkinsonian cell, whatever the case may be. So those tools will enable, you know, today maybe we're talking autologous, but there's already people looking at allogeneic therapies for CAR Ts. Pretty challenging. I mean, we're not there yet, but, you know, given the pace that, you know, that we could be going, and we will be going, I mean, it's not going to stop. I think there's just going to be tremendous opportunities to edit cells and just make customized products for patients. So that's my answer.
Yeah, so, you know, I would like to, you know, your question, as if I understood it correct, you know, what kind of genome would you create to make, you know, smart people? And I would like to have a genome edited so I just eat veggies and go to the gym every day. So, and you don't age. So I think it's a very, you know, I think as Bernard pointed out, the genome sequencing and knowing more about genome, I say, is this what we really want? I think there's a beautiful book from Siddharth Mukherjee that just came out. It's on New York Times, but it's one of the best on the bestseller list. And he asked the same question. You know, do we really need to know everything? Okay, even if we know everything, what are we going to do? And I think we are in exciting times, but I think we have to, you know, it's like an iPhone. You can be on the iPhone all day, but do you really want to be on the iPhone all day? I think we have to prohibit ourselves. How much do we want to know? And I think that is being smart. Smart is not trying to do everything that is available there. And I think we, as you know, CRISPR technologies, and you can edit anything and create multiple, you know, clonal types that you want. But you don't want to do that because that is, you know, I just heard that somebody is cloning humans in Italy. And possible because the technology is there. We could have Dolly the clone there from Italy now. But I think we as, you know, 21st century people should be really smart of what should we use and what shouldn't we use. I think it trickles down not only into science, but into every aspect of our life. And I think that is the key. And it, you know, genome, I'm very skeptical about genome sequencing because you don't want to know that you will get sick at 55 and then you can't do anything. I think I would take it with a little bit of caution.
Bernard, would you like to comment on this?
Yeah, I think most people would have few ethical concerns about editing out of the genome, like, for example, the Mendelian mutation that cause terrible rare diseases. We know there's thousands of those. So I think you could get a consensus on that it'd be a good thing to do. If you start doing that, and I think society, you know, because there's an economic impact, you know, the cost of all those rare diseases that affects 30 million Americans is non-trivial. So I think there will be, you know, a push to get that done sooner or later. I think the tough question will come after what comes next after that. Well, you know, do you want to edit out the genes responsible for, you know, obesity or, I mean, that would be a lot more difficult to do because you get so, you know, you're now dealing with networks. But, you know, there is some tough questions like beauty, for example. You know, if we somehow acquire the capability down the road to make people, you know, more muscular or more, preacher, or, if you can control their hair color, or if you can make them smarter. Do we want to do that? I think you'll probably have a very broad range of opinion about that, but I think ultimately it'll get done. There'll always be people wanting to do that, and where that will lead, I don't know. I'm not sure I want to think too much about it, but it's a question that as a society we'll have to deal with.
Yeah, and so I think these tools are making them available to us as a society, and what you raise is the question about what do we as a society do, and it's not just Western, it really is a global issue because these are globally available and research. And these questions is going on around the world, and different societies may have different, as well as different individuals have different views of what's acceptable or not. But I think that's probably a good question to end on because that's probably about one of the biggest questions that we've tackled so far. And one way to answer those big questions is really to talk about it further over drinks, which is actually a well-known solution to many problems that's been well explored over centuries. Join me in thanking the panel.